Hi, my name is Suzuki, and I'm a PhD student at the Center for Ocean Life at the Technical University of Denmark. And I will talk to you about the mechanisms and the fluid dynamics of foraging in heterotrophic nanoflagellates. Heterotrophic nanoflagellates are unicellular microorganisms that can range from 2 to 20 micrometers in diameter. They're ubiquitous in aquatic environments, and they're main grazers of the bacteria and picophytoplankton, giving them a key role in the microbial loop or in other biochemical cycles in the waters. And they are characterized by possessing 1 to 2 flagella that serves them for swimming or to handle or capture prey. Being so small, though, presents them some feeding challenges. First of all, they live in the ocean can be a nutritionally diluted environment. However, their clearance rate to face predation mortality is estimated to be 10 to the 6 times their own cell volume per day. And moreover, they living in this low Reynolds number regime means that that the water becomes viscous, and it's a physical impediment for predator-prey contacts. So how do the flagellates overcome these impeding effects of viscosity? Well, they create feeding currents by beating their flagellum that has hairs or mastigonines. And while they beat their flagellum, they create these flow fields towards a cell body, and any potential prey embedded in this flow field can be captured and handled for ingestion. The prey capture process in flagellates can be divided in a number of steps. First of all, the searching time is when the flagellate is creating this feeding current to be able to, and waiting for a prey contact. Upon prey contact, the flagellate can change its flagellum beating behavior to be able to capture the prey and either ingest it or reject it. Once it's ingested or rejected the prey, then it returns to its searching mode. During the time that it's handling the prey, this is called the handling time or the handling period. And the handling time is exclusively for flagellates that will not encounter any other particle while handling this prey. For this project, we have studied the prey capture process of 4 different species of flagellates that I will describe now. First of all, Pteridomonas danica attaches to the surface by means of a stalk, and is characterized by having a crown of tentacles around the beating flagellum that beats that continuously beats and creating this feeding current towards these tentacles, and then the prey will be captured by the tentacles and slowly dragged towards the body to be phagocytized. Pteridomonas danica does not change its flagellum behavior when it's captured a prey. It continuously creates a feeding current, meaning that it can be handling a prey while also searching at the same time. Paraphysomonas foraminifera attaches to the surface by some plasma thread and has 2 flagella. One that is long and beating, creating the feeding current, and the other one remains inactive while it's searching. When it enters in contact with a prey, the long flagellum hooks over and starts beating at a higher frequency, and the shorter flagellum also becomes active, and they're like interacting the 2 flagellum, and by the interaction of these 2 flagella, they retain the particle. And in this case, if the flagellate decides to reject the prey, it will simply return its long flagellum to the normal position and release the prey to the environment. However, if it wants to ingest it, the smaller part, the shorter flagellum will push the prey towards the cell body, and then it will be phagocytized. Here we have an example of an observation under the microscope of Paraphysomonas capturing and ingesting a bacteria. Cafeteriero emergencies attaches to the surface with its posterior flagellum while it creates a feeding current with its anterior flagellum. When a prey enters in contact, is captured, the particle enters in contact straight on the cell body, and the short flagellum stops beating and hooks over and physically retains the particle between the flagellum and the cell. To release it, if it decides to reject the prey, it just releases the particle by simply returning to its original beating position. And if the particle is ingested, it adjusts while it's being captured, and then it returns again to its searching mode. Here we have cafeteria capturing a particle. Sometimes it continues beating because the prey has to be readjusted the position to a more optimal position for phagocytosis. And there you go. Now it's starting at phagocytosis. The short flagellum, the short beating flagellum has stopped, and it's retaining the particle while it's being phagocytized. And then it'll return to searching mode. And lastly, we have pseudobodo. Pseudobodo attaches to the surface with its long flagellum and with its posterior flagellum, and is characterized by having a long flagellum in anterior flagellum that is looped helically in front of it, creating this feeding current towards the body. When it captures prey, the prey enters first in contact with the flagellum, and it travels along the flagellum that is beating really quickly. The beating frequency increases and is retained between the cell and the flagellum for a moment. And then if it decides to reject the particle, then it'll simply return to the searching mode, and the particle will be released. Or either it can also ingest the particle while being captured, and then it'll return to searching mode too. Here we have an example under the microscope. Now it will increase its beating frequency to be able to capture the particle and ingest it, and now it returns to its normal searching mode. Pseudobodo is also characterized by having a second mode of rejection. I have called it the lash rejection because it's actually when it entangles its long flagellum to physically push away the particle. So once it's captured, it may decide that it doesn't want it, and then it'll physically be pushing it away until it's out of contact, and then it'll return to its searching flagellar behavior. Here we have it's captured a particle, but it decides to reject it. So now it untangles its helical long flagellum, and it's physically pushing away the particle until it's out of reach, and then it's able to return to its searching mode. Handling times can be the upper limit for ingestion rates. So therefore, we have investigated the handling times of Paraphysomonas, cafeteria, and pseudobodo. 20 events were measured for ingestions, and 20 rejections and 20 lash rejections were studied. As you can see, ingestions in general are the cafeteria had longer ingestion handling times, and then Paraphysomonas had a bit more shorter handling times for ingestions, and pseudobodo had surprisingly very short periods of handling times for ingestions. Rejections in general are all short, except pseudobodo, the characteristic lash rejection would take longer because once uncoiled, the flagellum actually has to return to, it slowly returns to its searching mode, so that takes a bit longer time. We can calculate the maximum ingestion rates with these handling times, and they range from 1,000 to 20,000 prey per day. This is largely in agreement with results from incubation experiments that can range from 600 to 6,000 prey per day ingestion rates. But if we also take into account the fact that there's a lot of cases that of particles being rejected, that the rejections also take time, even though they're shorter, then that can actually adjust them more to what is being observed in experiments. So therefore, we can mechanistically understand now with these handling times that we see that we have measured, they can be, they agree well with the results within incubation experiments. So manual particle tracking was also performed on 2 individuals of each of the species in the study. And here we have an example of an individual of Pteridomonas danica. Particles here in green were the ones that were captured by the flagellates, and the ones in red weren't. From calculating the velocity component Vz, so the one that is perpendicular to the surface, we're able to obtain a clearance rate. The clearance rates are the volume of water that can be cleared out or searched through by the predator for potential prey captures or prey encounters in a certain period of time. So in order to estimate the clearance rates, a theoretical clearance disk was positioned in front of the flagellate, and the flow rate that goes through it was calculated. The diameter of this of the clearance disk was set to contain all the tracks that were captured or that strongly interacted with the flagellum. And now to know the overall force that this beating flagellum is delivering to be able to clear out large volumes of water, we applied a point force model. The organism is represented by a simple force, point force of magnitude F that is located at a height h from the surface. And then this model also includes 2 parameters from the clearance disk. That would be A, the radius of the clearance disk, and D, the height of this clearance disk from the surface. And since we're working in a low Reynolds number regime, the governing equations are the Stokes equations for the momentum conservation and the mass conservation, from which Blake derived a solution for a flow due to a point force with the presence of a wall. And then Roth et al. Also then derived this last equation here to calculate the flow rate through the green disk, where F represents the action of the flagellum here. A is the radius of the disk, and D is the height of the disk, and H is the height of the force. For the case of the studied flagellates, the unknown parameter is Q, is F, is the magnitude of the force, and which we can calculate with the given clearance rates that we obtained through particle tracking. Okay, and here you can see the resulting particle tracking from one case of each species. And the group of the 4 graphs here on the right are the 2 species that create feeding currents that are perpendicular to the surface. And they're compared to the theoretical flows that are represented generated by the calculated force, magnitude of force from the model. And you can see that it represents well, that the model is representing well the actual observed flow fields. And on the right here, we have also 2 cases of one of pseudobotha and one of Cafeteria rhombergensis. And these are 2 individuals that are creating feeding currents that are parallel to the surface though. And note that due to the fact that here the rotational symmetry was not possible to be assumed In the case of the parallel flow fields, as it was assumed in the perpendicular flow fields, the calculation for the forces were estimated with a distinct approximation than the one explained before. All these organisms here sit at different at the surface at different heights and are generating these feeding currents with different flagellar behaviors. However, they're all creating this flow field with hourglass-shaped streamlines that are characteristic in the low Reynolds number regime. And here we have the resulting volume-specific clearance rates for the 8 individuals. They all range from 10 to the 6 to 10 to the 7 cell volumes per day, which is agrees with some of the results given that incubation experiments have given. And then the forces that were estimated are also on the range of 3 to 13 piconewtons, and this is also agrees with other other studies that have also attempted to estimate the force being generated by the flagellum. Obviously, the point force model is not taking into account the presence of the cell nor of the flagellum. So therefore, we expect that the force would actually the force required to generate these feeding currents would actually be a bit more larger. And in conclusion, we have that, we've observed that flagellates definitely present distinct forms of predation. Their flagellar behaviors vary a lot from species to species. Ingestion rates may be limited by the handling times, and the handling times that we measured give maximum ingestion rates that largely agree with incubation experiments. Clearance rates estimated with particle tracking were also in the order of 10 to the 6 cell volumes per day, which is the magnitude of estimated for their population success and facing predation mortality, and is also agreement with other incubation experiments, and also that the point force model has allowed us to calculate this force of value of reasonable magnitude for feeding flows perpendicular to the surface and also parallel to the surface. Thank you very much to my 2 supervisors, Anders and Thomas, and also thank you for your attention.